So I'm just going to give our speaker a brief introduction today. He's Dr. Nathan Burkett-Cadena. I met him at the Utah Mosquito Abatement Association meeting about a month and a half ago in late October, where he gave a talk that I thought was pretty appropriate for what we were trying to do, which was to talk about Oropouche virus. So he's joining us today from the University of Florida Medical Entomology Laboratory. So he's located in Vero Beach, Florida. He's an associate professor down there, and he's a vector-borne disease ecologist with interest in host preferences and how those preferences interact and influence arboviral disease transmission. If I get any of this wrong, Nathan, you'll have a chance to correct me. Today, he'll be giving us, like I mentioned, kind of an overview of just kind of where we're at currently with current knowledge of Oropouche, including the disease ecology. And like I said, I thought it was a pretty good talk at the Utah meeting. I'd see a few of you here were also there, so you might have a little bit of overlap, but hopefully it can at least freshen you up a little bit. And then we'll follow that. He'll talk for maybe about a half hour, a little bit longer, and we'll follow that up with some brief notes from Dr. Chris Roundy, who's the medical entomologist for the CDPHE, Colorado Department of Public Health and Environment. So he's going to speak to a few local kind of things. There's not too much to know so far, so that'll be some brief comments, then we'll open it up for question and answer. So without further ado, the floor is yours, Nathan.
Okay, can you guys see my screen?
Yeah, looking good.
Let's see, I think I have to put this in. All right, does that look okay?
Yep.
Can you hear me all right? Okay. All right, well, thank you so much, Tim, for the invitation to speak today. I think this virus is pretty new on most of our horizons. So fairly new to mine, although I have been working with mosquitoes and culicoides both for a number of years now. So I've been very interested in this virus for quite some time. So I never thought that I would get the chance to talk about it until this year. So real surprising. All right, so I'm going to start off with this. This is a Florida weekly arbovirus surveillance report that is free to anyone, I believe, in the world to get. You just have to email the coordinator for this. Her name's Andrea Morrison, and they send out these reports on arbovirus activity in Florida. And so there are really valuable data on like sentinel chicken seroconversion to viruses like West Nile virus, St. Louis encephalitis virus, and Eastern equine encephalitis virus. So these zoonotic mosquito-borne viruses, and gives everything about the number of cases in positive humans, asymptomatic donors, wild birds, domestic birds, like I said, sentinel chickens, how many counties have reported in that week, and the yearly totals. These reports also have data on exotic mosquito-borne anthrapanoses like dengue virus, chikungunya virus, and Zika virus, and they break it down into travel-associated cases and locally transmitted or locally acquired cases in Florida. And you can see that it was a hot year for dengue with 824 travel-associated dengue cases as of the end of October, and 70 cases of locally acquired dengue. And then this year, a new category was Oropouche fever. So in Florida, we had 90 travel-associated Oropouche fever cases documented in Florida. So this is where all this started. So Oropouche virus, as you guys probably know, discovered in Trinidad in 1955. It was isolated from a human being, from a young male forest worker who presented to the local clinic with febrile illness, backache, cough, and fever, which he recovered from. Well, the arbovirologist there isolated the virus, and they named it Oropouche virus after Oropouche Meadow, which is a municipality in northern Trinidad. And I say Oropouche because I have a friend from Trinidad, and they told me that in Trinidad they say Oropouche, so that's what I say. All right, so the virus then was not seen again in Trinidad, but it popped up in Brazil just a few years later in 1961. The first human epidemic was documented in the state of Pará. And so this is a state that surrounds the mouth of the Amazon River. There are estimated 11,000 Oropouche virus cases. And then there were sporadic and periodic epidemics of Oropouche virus in this Amazon region of Brazil in the states of Amazonas and Pará. And they estimated at least 250,000 human cases of the virus. Well, over the next few decades, there were cases documented in neighboring countries like Peru, Ecuador, Colombia, and Panama. And so this virus is thought to be fairly widespread in its natural distribution. Well, then of great concern, Oropouche virus was detected in Haiti in a child with acute febrile illness. And then of really great concern was the outbreak this year in Cuba. And in Cuba, the cases were distributed basically across the entire island. So all the way from Havana province in the west to Santiago de Cuba province in the east. And these are definitely not the only cases that occurred, but these were the cases that were documented at the time of publication of Benitez et al. All right. So, as I said, we had a bunch of 90 total travel-related cases in Florida, and all of these had documented travel to Cuba. Well, a lot of these cases happened in our larger cities, or were at least detected in our larger cities, like Hillsborough County, where Tampa is, and then Jacksonville, Orlando, Palm Beach, Fort Lauderdale, and Miami. And so I think it's expected that, there were international travel between citizens of these cities and Cuba. But then we also saw a fairly large number of cases from smaller counties, not smaller counties, but counties that do not have the same level of international travel as you would expect from, say, Miami, Tampa, and Jacksonville. And so a interagency task force was established to try and determine what were the risk of local transmission of Oropouche virus in Florida. And the questions that they kept coming back with are which arthropod species can transmit the virus and what vertebrates could serve as hosts for the virus, in the eventuality of a locally transmitted case. All right. And then I think, the big question is, will this virus establish in Florida? I'm thinking from a Florida perspective, but, could it establish elsewhere in the United States as well? All right. So to understand the virus, you have to understand, I think, its origins. So Oropouche virus is a member of the Peribuniaviridae. It's an orthobunyavirus. And the very first or prototype orthobunyavirus was described from Uganda in this southwestern part of the country, which is home to the Rwenzori Mountains. And this is the place where the mountain gorillas are found. And so this is a very wild cradle of arboviruses, a very interesting place. And this is where the name bunyavirus comes from the village of Bunyamwera, Uganda. All right. So there are several orthobunyaviruses that occur in North America, which we can look at to try to understand Oropouche virus. This includes the California encephalitis virus complex, La Crosse virus, Jamestown Canyon virus, snowshoe hare virus, and keystone virus. So depending on where you live, you may be more familiar with one of these than the others. But the vectors of these viruses are mosquitoes of the genus Aedes, and in some cases, mosquitoes of the genus Culicida. And the natural hosts of these orthobunyaviruses are mammals for the most part. So everything's from squirrels and chipmunks to slightly larger rabbits and hares, all the way up to deer. So when the first outbreaks of Oropouche virus were happening in Brazil, in order to inform vector control and interrupt transmission, they really wanted to understand what were the vectors and the natural hosts of Oropouche virus. So a number of outbreaks happened in these villages on the southern side of the Amazon River. in this greater Amazon region. And this map shows the some of these towns where investigators were trying to determine the vectors and hosts of Oropouche virus. And what they did to look at the vectors was trap a bunch of mainly mosquitoes. And I think this is probably because the known vectors of other orthobunyaviruses were mosquitoes. So Culex quinquefasciatus was the most common mosquito trapped and screened for the virus in the first 6 or so years of investigating these outbreaks. And they isolated Oropouche virus on 3 occasions, or from 3 pools of Culex quinquefasciatus during these studies. And then at some point, they started trapping additional potential vector insects. And so in the 1970s, they added black flies and ceratopagonids to their captures and to their testing. So they weren't testing these in the 1960s. But in the 1970s, they captured Culicoides paraensis in very large numbers in Santarém, Brazil, and they got 4 isolations of Oropouche virus from this species of ceratopagonid. So in total, they had 3 isolations from Culex quinquefasciatus and 4 from Culicoides paraensis, so rather equivalent and a very similar infection rate in these 2 different organisms. So then they wanted to the same group of researchers wanted to determine whether these 2 different insects could transmit. So they first evaluated the vector competence of Culex quinquefasciatus by letting it feed on a hamster infected with Oropouche virus with very high titers of the virus, and they only had 1 positive mosquito out of all of the mosquitoes, say 200 or so female mosquitoes that they tested, which showed that Culex quinquefasciatus is not an efficient vector of this virus in the laboratory. So really only a 0.5% transmission rate. Well, when they looked at Culicoides paraensis, they found it between 13, 15, 44, and 50% of Culicoides paraensis could actually transmit Oropouche virus by bite after they'd fed on the blood of an infected human being. So this showed them that this was the first conclusive evidence of transmission of an arbovirus of public health importance to man by a member of the family Ceratopagonidae. This was the very first time that a Culicoides had ever been shown to transmit a pathogen of human health importance. All right, so we have to talk a little bit about Culicoides and what they are. They are tiny, biting or blood-sucking flies in the order Diptera. They are members of the family Ceratopagonidae. They are called biting midges. In Florida, we mainly call them no-see-ums or sandflies or sand gnats even. And I say they are not mosquitoes because this does not seem to be common knowledge. In fact, they are not even really closely related to mosquitoes. They're more closely related to black flies than they are to mosquitoes. And mosquitoes are more closely related to chaeoborids or phantom midges and frog-biting midges than they are to ceratopagonids. All right. In fact, the family Ceratopagonidae is not mainly even composed of blood feeders. So Ceratopagonidae is mainly composed of predators of other insects, nectar feeders, and parasites of other insects. So most genera and species of ceratopagonids do not bite. I'll just show you a couple of examples of some interesting ceratopagonids. So the stick ticks. So these are ceratopagonids that are parasitic on walking sticks in the tropics. This is one that was in Colombia. It's got 1, 2, 3, at least 4 ceratopagonids that are living as ectoparasites on its body. This is the genus Forsythomia. And here's one in Florida that is not a long-term parasite, but it is a feeder on walking sticks. So graduate student captured a walking stick and noticed something on its rear end, brought it to me, and I was able to get this photo of a Forsythomia biting walking stick. So the genus Culicoides is really diverse. There's over 1,300 species in the world. I think there's probably over 200 species in the US, but I can't get a really good count on this. I can say with some pretty good certainty that we've got 50 species in the state of Florida. So their life cycle is very similar to that of a mosquito. The females feed on blood in order to produce their eggs. The males do not bite. Females lay their eggs in wet environments. Their larva is not very similar to that of a mosquito. It looks more like a wiggly little nematode than it does a mosquito larva. And their pupa also, to me, look quite different than a mosquito larva. They're much straighter, and they have a sort of a wavy motion rather than a tumbling motion. And after the pupa stage, of course, we have the adult. So similar to a mosquito, but a little bit different. And the habitats that the different Culicoides species use are very diverse as well. So like mosquitoes, each species is more or less adapted to a particular kind of larval habitat, like seepages or springs, marshes, damp tree holes, stream edges, wet pasture, or the edges of puddles. And so I've just put on this slide some of the species that are really common or of a veterinary importance in Florida. And I've circled the species Culicoides paraensis in the upper right because this species is known to breed in damp tree holes. All right, so let's talk about Culicoides paraensis for a minute. So it was described from specimen captured in the state of Pará, Brazil, and that's where its name comes from, Pará paraensis. All right. And this passage here from the book, The Sandflies or Culicoides of Florida, is about the larval habitat of this. And you'll see tree hole, tree hole, tree hole underlined several times, but you also see cacao pods and rotting calabash. So suggesting that there is some variability or flexibility in where this particular midge will breed. And so this is what a pile of rotting cacao husks look like. So on a chocolate or cacao farm, when they harvest these pods, cut them open, extract the seeds and roast them to produce chocolate, they discard the exterior of the pod called the husk. And this is a beautiful and perfect larval habitat for Culicoides paraensis. So you imagine an accumulation of cacao pods like this can produce extremely large numbers of any insect that would breed in that habitat. All right. So Culicoides paraensis is found in the damp or wet tree holes and mainly in deciduous trees, but it really is not found in artificial containers. So not in discarded tires, not in man-made containers, not in bird baths, and not in bromeliads. So a lot of our container inhabiting mosquitoes also breed in tree holes, and maybe that was their previous evolution or previous ancestral habitat. And they've adapted to artificial containers, but it doesn't seem like Culicoides paraensis has done that. All right, so what is its distribution? Well, it's found mainly throughout the eastern United States, going down into Central and much of South America, all the way down to northern Argentina. It is associated with forests, so we find it in the forested regions of eastern North America, Central America, and South America. All right. Thinking of the perspective of how likely it is to transmit Oropouche virus in Florida, this graph shows the frequency of abundance of about 25 different biting midge species that one of my graduate students, Kristen Sloyer, collected during her master's thesis. And I'll just put an arrow to paraensis to show that we only collected about 20 females of this particular species of biting midge during 2 years of studies and over 100,000 biting midges captured and identified. So at least based on the traps, it does not seem to be extremely common in Florida. Well, a study that was done in the Tennessee Valley, looking at the species of mosquitoes and no-see-ums coming to bite humans in the forest, was done by Snow. And he had his insect collectors sitting at 3 different heights in the forest, one at ground level, one at 30 feet, and one at 75 feet. And they stayed at these on platforms and collected all of the insects that came to bite them throughout the day and throughout the night. And what they found was that Culicoides paraensis was about 25 times more common at the 30-foot height than at the forest floor inside the forest. So that seems to indicate that this species of biting midge is really more active in the tree canopy than it is at the ground level. And they also found that Culicoides paraensis was about 75% less common at the forest edge than the forest interior. So that means that Culicoides paraensis, although it is found in the eastern United States, it really likes to be in the interior of the forest, and it also likes to be up high, basically in the treetops, probably in order to bite one of its favorite hosts, which appear to be squirrels. Now you can contrast that with a similar study that was not done at different elevations, but done throughout the day in Santarém, Brazil. And so this graph shows the number of bites of Culicoides paraensis captured on human bait. And at 6 p.m., the captured over 1,000 Culicoides paraensis coming to bite them during the study, over 1,000 in an hour. But if you look at the photo to the left, you can see the large number of discarded cacao pods, which probably were producing the very large, artificially large numbers of midges in those areas, which probably caused the outbreaks of the virus in those areas. All right. So in Florida, I would hazard to say that the distribution of this particular midge species is correlated highly with the percentage of forest cover. So in the northern half of the state is where we find Culicoides paraensis, and that's really where we have the majority of our forests. All right. So I would say it's not likely that if a traveler comes back to Tampa or Miami or Orlando or Jacksonville infected with Oropouche virus, that they're going to encounter Culicoides paraensis in their neighborhood. But what about the other 49 species of biting midge in Florida? Are they likely to transmit Culicoides Oropouche virus? And for that matter, what about the mosquitoes of Florida? Are any of the 90 or so mosquito species in our state likely to transmit Oropouche virus, or the over 200 species of mosquito that we have in North America? Well, to really understand this question, you have to look at the criteria for incriminating a vector species. And the World Health Organization has developed these criteria set forth to help determine whether a particular species could be important in transmission. And basically, all of these have to be satisfied in order for a species to be incriminated. So an association in time and space between the insect and disease and the host animals, suspected vectors collected under natural conditions harbor the pathogen, evidence that the suspected vector can transmit the virus from an infected host to an uninfected host in the laboratory. We've already talked about vector competence, and evidence of direct contact between the suspected vector species and host animals. So this last one is really important. So the suspected vector has to feed with pretty high frequency on the host animal. So what is the host animal of Oropouche virus? Well, the same studies by Pinheiro et al. In Brazil not only looked at the vectors, but they collected very large numbers of vertebrate animals, including mammals, birds, reptiles, and amphibians. You can see that there were at least 2,000 mammals that were sampled, over 11,000 birds, 8 reptiles, not amphibians, I guess. And Oropouche virus was only isolated from sloths. So despite testing 11,000 birds, they never recovered that virus from any birds. All right. So it seemed that the natural transmission cycle of Oropouche virus in South America involves sloths, and the Culicoides paraensis in the enzootic cycle. And this spills over to humans in an epidemic, but it is very likely that humans also contribute back virus to the vector in a peri-urban or urban cycle of the virus. And I think this is supported by this vector competent studies where they used humans as the source of virus to feed Culicoides paraensis for the vector competent studies. High titers of virus on the range of log 5 to log 7 were obtained from humans for these experiments. That's high virus titer. All right. So what do we know about sloths? Well, they're very tropical arboreal animals, of course, and they are really only found in the American tropics and extend into Central America only, really about as far north as Costa Rica. No matter which of the sloths you're talking about, the three-fingered sloths or the two-fingered sloths. And there are no sloths in Cuba, Haiti, or the USA. So what could be a wild host for Oropouche virus in the United States? I would say there is probably not one, but we do not know that for sure. All right. So I would say that the potential actual host animal for Oropouche virus in the USA is a human being. That if Oropouche virus is going to be transmitted locally, that humans are going to be the source of the virus and wildlife will not be involved in transmission. Okay, so to understand which potential vector species could transmit locally, we really need to understand which species frequently bite humans. I know we think that all mosquitoes and all no-see-ums bite humans, but actually most of them feed fairly infrequently on humans. So if you assume that, say, a mosquito or a biting midge survives long enough to feed 4 times in her life, then that, as a species, they would have to take 50% of their blood meals from humans to be a vector of Oropouche virus. They would, because they have to bite once to be able to acquire the virus, and a second time in a subsequent blood meal from the same host species to be able to transmit. So they'd have to take 50% of their blood meals from humans in order to be considered a possible vector of Oropouche virus. So blood meal analysis is a technique for determining what animals are bitten by wild blood-feeding arthropods. So this consists of collecting wild blood-engorged females, extracting DNA, using PCR to target vertebrate genes, sequencing of amplicons, and then comparing those sequences to genetic databases. So this has been done for many different mosquito and biting midge species throughout the world. And this is a focus of what we do in my laboratory to help understand, inform vector incrimination and also understand transmission of mosquito-borne viruses. So this graph condenses down all of the studies from my laboratory on blood meal analysis of the Culicoides species, which are common in Florida. And the different colors on this graph represent the different host classes. Blue is birds, yellow is mammals, green is amphibians, and black is reptiles. And the different Culicoides species are listed down the left side of the graph. And you can see that about 5 species feed primarily on birds. Most of the species tend to bite mammals. And then there are even a couple of Culicoides species which seem to like amphibians or will also feed upon reptiles. However, when we look at the percentage of blood meals from human beings, none of the Culicoides species that we've examined have ever approached 50%. So you see the arrow pointing to Culicoides paraensis, takes 20% of its blood meals from human beings. So it, in Florida, at least, is not approaching a level which would classify it as a potential vector of Oropouche virus. Similarly, when we look at the mosquitoes, we see that the mosquito species feed on a diverse array of hosts. Some of them, particularly species of the genera Culex, Culicida, and Dinocerides, seem to prefer to feed on birds, while a number of Aedes, Wyomia, Saurophora, and Anopheles prefer to feed on mammals, while a few species prefer to feed on reptiles and amphibians. If we take these same mosquito species and we rank them in their utilization of human beings as hosts, we see that only 2 species take more than 50% of their blood meals from human beings. That's Aedes aegypti and Aedes albopictus. These are well-known and well-characterized vectors of anthraponotic mosquito-borne viruses. So if only 2 to 3 mosquito species in Florida and maybe the entirety of the United States are likely to transmit Oropouche virus, and that is if they are biologically capable. So what do we know about the vector competence of these mosquitoes for Oropouche virus? So not a lot has been done with North American mosquitoes, but my former PhD student, Bethany McGregor, performed a series of experiments when she was on a fellowship at the CDC looking at vector competence of Culex tarsalis, Culex quinquefasciatus, and Culicoides sonorensis for the original 1955 isolate of Oropouche virus, which was obtained from the blood of a forest worker in Trinidad. And what she found was that none of the Culex tarsalis could transmit Oropouche virus. Less than 1% of Culex quinquefasciatus could transmit Oropouche virus, very similar to the findings of Pinheiro et al. But for Culicoides sonorensis, nearly 20% of the females that were tested could transmit. So there does seem to be some capacity for a Culicoides species to transmit Oropouche virus in the United States. So what do we know about Culicoides sonorensis? Well, it is a very, very important vector of hemorrhagic disease viruses that affect cattle and sheep and deer in North America. These viruses are epizootic hemorrhagic disease virus and blue tongue virus, and these are often fatal for these animals. But Culicoides sonorensis rarely, if ever, bites humans. So there's only a couple of studies which have ever documented the host use of Culicoides sonorensis in North America, and there were no human blood meals found for this particular biting midge species. So if it's not biting human beings, and cattle, sheep, and deer are not natural hosts for the virus, then Culicoides sonorensis probably cannot be involved in transmission of Oropouche virus. All right, so getting back to this question, will there be local transmission of Oropouche virus in the US? I left my crystal ball in my other pants, so I can't tell you for sure, but I think that it is possible, but not very likely. So our mosquitoes appear to be poor vectors of Oropouche virus. Our Culicoides do not feed commonly on humans. And so I would say that establishment of Oropouche virus in the US is unlikely, but that imported cases may result in sporadic local transmission of the virus, similar to what we see with dengue virus here in the USA. All right, so I'll just close with this video, if I can play it, that my son took of a sloth scratching itself in Panama. And I always wondered were those Culicoides paraensis bites that it was scratching. And that is all I've got. Thank you so much.
Thank you, Nathan. I think we'll hold off on, if you've got some questions, hold on to them or throw them in the chat. Let's just jump over to Chris Roundy real quick and see if he's got a little bit more information to follow up from that.
All right, thanks, Tim, and thank you for that presentation, Nathan. That's really helpful information. Let me see if I can share the right screen here. Okay, so I just have one slide to present today, and all of this in the context of what we just learned about all of these viruses. This is from a CDC EID report that came out last month, showing the locality of possible Oropouche vectors throughout the US. So if we look at our region specifically, I should also say the most likely vectors start with A, working our way down to C based on the vector competence data. So the most likely vector, Culicordes paraensis, we do have in Colorado, also can be found in Nebraska. If we take a closer look at where this is found, as was mentioned, it tends to not be in our urban environments. I believe in Colorado, this has been primarily found in agricultural areas. However, if Culex quincefasciatus or other Culex species are found to be possible vectors, then we have more states throughout the region that may need to be concerned. These are also more common and can be found closer to urban areas than our Culicoides species. And then lastly, Culicoides sonorensis can also be found in a few states in our region. However, like the other Culicoides species, tends not to be in urban areas and is not very common or widespread in the states where it can be found. Also wanted to mention here that while the vast majority of travel-associated cases have been in Florida, a few other states have seen travel-associated cases, including California, Colorado, Kentucky, and New York. And that is one case in each of those states. So we have had one travel-associated case here in Colorado. So overall, we are kind of anxiously awaiting further vector competence studies using the current outbreak strain, seeing if that changes any of the vector competence data that we have historically. And based on that, we will change our plans for, what we're looking for, what we're testing, and how we approach our vector season next year. That is all I have locally. It's more questions than answers at this point, unfortunately. So I will turn it back to you, Tim, for questions.
Yeah, and I think I mentioned earlier, unfortunately, this talk takes place at the same time as the biweekly CDC Oropouche dengue virus update call that they hold on Fridays. If you're interested in attending those, maybe reach out to me. I can kind of put you in touch with the right person. But one thing I've noticed being on those calls is it's kind of the same thing as Chris mentioned, a lot of kind of guesswork and sort of they're doing some vector competency studies, but the results are slow to trickle in. And as we learn more from that, then we'll be able to say a little bit more about some of this stuff. But currently, it is a lot of kind of projections and estimations and stuff, which I think both of you did a great job of kind of outlining for Florida and then for our local area as well. So yeah, if anybody has any questions, now is the time. It can be about the topic today. If you have maybe some suggestions for stuff you'd like to see going forward. I know we had heard a few things last time we held this a month, 2 months ago, which we're keeping in mind for the spring. We're planning to maybe hold 2 or 3 of these in the spring while people are a little less busy before the summer. And then in the summer, we'll maybe suspend them or maybe transition to like a shorter kind of update type of call where we update on the local kind of situation in the region here. So a little undecided, but we're happy to take feedback on that as well. And if you'd like to see something about a certain pathogen or something of interest, then yeah, of course, let me know. So yeah, if you have any questions, just feel free to unmute yourself or put it in the chat or whatever, and go from there. If not, and I think, to be honest, there's not too much to be questions besides these broad ones that we're all waiting for answers for. But if you don't have any questions, then I think we can head out. I do want to give everybody a chance, but if you want to ask something later or follow up, then you can get in touch with me again. I can put you in touch with Nathan or Chris or any of that stuff. I put our website on in the chat there, and then also we have yeah, there's a contact email on there you can find. So otherwise, I will see you next time. That should be probably maybe later in January or something like that, and we'll get the information out as best we can. So yeah, thanks again, Nathan. Great to see you. Great to hear again. And,
thank you. Thank you. All right. Take care, guys.
Yep. Bye, everybody.